Hello and welcome to Biology Interest. Today we're going to be looking at some more A-level biology material, and in this video we're going to be looking at the nerve impulse down an axon. So what I have right here is a diagram of an axon with some voltage-gated channel proteins inside of it, and we're going to go along each step of a nerve impulse down the axon. So there's two important things that we need to be able to see in this diagram, and that's the two types of ions I'm going to be looking at, which are important in the passage of an action potential. So I've got red sodium ions, which are going to be in red, and their voltage-gated channel proteins are also going to be red. And we've got green potassium ions and potassium voltage-gated channel proteins. So just keep in mind which colour is which. So let's get started. So normally when a axon is not being stimulated, it's kept at a resting potential, and it's kept this way by essentially having both the sodium voltage-gated channel proteins and the potassium voltage-gated channel proteins closed, so the ions can't easily diffuse in or out of the membrane. While it's like that, we also have a active transport protein, a sodium-potassium pump essentially, which is using ATP to transport sodium out and potassium in to keep the concentrations balanced and ready for an action when an action potential arrives. So now we've gone over what currently is going on at a resting potential. A stimulus has occurred and we want to create an action potential. And so what essentially happens is the electrical impulse causes, from a generator potential, which we'll look at in another video, causes an action potential to begin. And this causes sodium voltage-gated channel proteins, shown here, which are what these are, to open. And so the sodium one opens first when we want to start the action potential. And this essentially causes sodium ions to diffuse rapidly from the outside of the axon to the inside of the axon. So we've got lots of them inside the axon now. And that essentially, because they're positively charged, increases the level of the positive charge inside of the axon compared to the outside of the axon. We call this potential difference. And so when an action potential occurs, the amount of sodium ions inside of the membrane compared to the outside, as well as the potassium ions, they cause the charge to go to plus 40, where before it was minus 65 compared to the outside of the membrane. So now the potential difference is higher inside the membrane, the axon membrane, than outside. And we call this the action potential. It reaches around plus 40, which is what we'll look at later in terms of why that's important. So once we've reached that plus 40 potential difference inside, it's a high enough charge to essentially trigger these voltage-gated sodium channel proteins to open in a nearby part of the membrane. And so that's how the nerve impulse travels down the axon. Every time one part of the axon, the potential difference inside reaches high enough, it triggers the next part of the axon, triggers those sodium voltage-gated channel proteins to open and allow the sodium ions in. And this continues all the way down the axon until it reaches a synapse, etc., or an effector. So now we're at this part of the membrane where the action potential has occurred and triggered the next one, and we want to reset it almost to allow another impulse to come along and begin it all at the action potential again. And so what essentially happens now is that the sodium voltage-gated channel protein closes and a potassium voltage-gated channel protein opens. And this allows the potassium voltage-gated channel proteins and the potassium ions to essentially diffuse rapidly out of the inside of the membrane to the outside of the membrane. And as we looked at earlier in terms of potential difference, this now causes the outside of the membrane to be have a higher charge or higher potential difference than the inside of the membrane. And in fact, so many potassium ions go outside that the potential difference actually decreases further than minus 65, which is what the resting potential occurs. And this is called a hyper-polarization or refractory period phase, which we'll look at later in why it's important. And at this time, after that occurs, to rebalance this hyper-polarization just to a resting potential level, we have a sodium-potassium pump occurring to pump lots of sodium out and less potassium in to kind of rebalance the concentration gradient, ready for the next action potential to come along and trigger an action potential. So hopefully that's looked at briefly how the impulse travels along and what sodium voltage gated channel proteins and potassium voltage gated channel proteins are open when and what happens. We'll just look at some other stuff which is important and you need to know to understand a nerve impulse. So this is quite a good diagram here of what happens. So we have step one here, which is what the resting potential is like, and what the potential difference is like normally. So normally it's more negative inside the axon than outside of the axon at around minus 65 potential difference, or millivolts. And so what happens here, what begins to happen here at step two, is that the action potential has been triggered by a receptor, and this essentially causes the sodium voltage gated channel proteins to open, causing them to sodium ions to rapidly diffuse inside of the axon, increasing the potential difference to around plus 40, which is important later. And so once the action potential has occurred in this part of the membrane and triggered more action potentials further down the membrane, it actually causes the sodium voltage gated channel proteins to close and the potassium voltage gated channel proteins to open, which causes the potassium ions to rapidly diffuse out of the axon and decreases the potential difference, so it becomes more negative inside of the axon. And here's our bit of hyperpolarization, step 4, where more potassium ions actually diffuse out than we want, and this decreases it further than minus 65, which is important later and is called the refractory period. And then we have the sodium potassium pump, essentially to rebalance it back up to an action potential, a resting potential level of minus 65. So there's two kind of terms that are important to know for this, for a nerve impulse, and that's the all or nothing impulse, as we can see here, and that's essentially that the action potential, the potential difference, must reach around plus 40, or the sodium voltage gated channel proteins in the nearby part of the membrane won't open and continue that nerve impulse. And this essentially means that a threshold level must be reached when you have an action potential, and it stops loads of tiny little nerve impulses essentially being created when we don't want them to be. And we also have a thing called the refractory period, which I've talked about just a bit before, and that's this hyperpolarization bit. And this is quite important because it essentially allows kind of three different things to occur. It means that the action potentials can only go one way. They can only go along the direction from the beginning to the end, because going forward, the membrane is at the resting potential, and so the action potential is high enough to cause an all or nothing response. While going backwards, the potential difference is actually too low, so when the action potential occurs, it doesn't reach that threshold potential, and so it can't go backwards, which is good because you only want the nerve impulse to go one way. It also allows discrete impulses because there has to be a gap between each one because of the refractory period. And so that means you don't have just one massive wall of impulse, and it allows the body to work out how big a nerve impulse is, essentially, how important this response is. If you've got loads of really quick nerve impulses, it's a big response, while if there's gaps between them, it's less of a big response, and it tells the body how much it needs to respond. And it also limits the number of action potentials because there has to be gaps between them. So there's also something to do with the speed of the impulse, which is the final thing I'm going to talk about. There's three main ways you can look at the speed of an impulse: increase the speed or decrease the speed. And that's a myelin sheath. And so you have the myelin sheaths, and then you have the node of Ranvier's, and it essentially allows saltatory conduction. So the impulse jumps between each node, and so that essentially decreases the amount of axon membrane that has to be polarized as the nerve impulse goes down, so it increases the speed of it. You also got a diameter, so the larger diameter, the better the surface area to volume ratio, the less ions that leak out, and so the faster the diffusion into the membrane and the better the speed of the impulse is. And you also got temperature, because an increase in temperature increases the rate of diffusion, so the higher the temperature, the faster the speed of the nerve impulse. So I hope this has been a pretty good overview of how a nerve impulse works. Please like and subscribe. Stay tuned for more videos from biology interest. Thank you.